Well, welcome everybody. I'm happy to be here and to introduce our speaker for today and the workshop presented by Cheryl McCutchan, 5 Simple Steps for Creating Research Diagrams. So I'm really a terrible artist. People kind of make fun of me in my family. Nobody wants to be on my team with Pictionary. And I'm equally bad when it comes to drawing scientific diagrams. I sit there with PowerPoint and I put together these really sketchy, sort of struggly diagrams, and I haven't taken the time to learn how to make good diagrams, mainly because I kind of pawn it off on my students and postdocs and rely on them to make the diagrams for our grants and our papers. And one of these days I'll find the time to actually learn, and I hope to learn something from this presentation as well. But when I do teach students in my first year course, PhD Fundamentals, I emphasize the importance of professional looking figures. When you write a fellowship application, for example, having a good model figure gives the reviewers confidence that when you submit a paper to a good journal or a review article, that the editors and reviewers of those journals are going to be more likely to accept your work. And the same goes when designing figures with data, is that having clear, easy to read, professionally presented figures with the right colors or not colors and all that makes a big difference as to how your research is viewed and how your thoughts are viewed in the science that you present. And that also goes for presentations, oral presentations. So it's really timely that we have this workshop, especially since many of you are also participating in the fellowship writing workshop. And I thank Harinder and Joanne Lee for developing it. And so with that, I'll hand it over to Joanne, who herself is a very accomplished artist and has started this art and science initiative. So thank you all, and I'll give it over to Joanne.
Sure, thank you, David. Did you want to say anything, Harinder, before I introduce Cheryl?
Oh, yeah. I'll quickly say that for all the I won't take too long. I just wanted to tell attendees this is part of our new career cohorts program we launched. So this is one of the parts where if people are interested in art as a career, or they want to have art in science, or they want to do art which is inspired by science, this is the career cohort for you. And we launched this. This is our first event of that cohort. So please feel free to participate actively, and there'll be a lot more events coming. Joanne will be the contact person, and she will take care of you very well. Thank you, Joanne, for taking lead on this, and thank you, Cheryl, for coming. Appreciate
You're welcome. Sure, it's an honor. So my name is Joanne. I'm the multimedia director of GPSTEM. You see all the podcast video stuff. I sort of have a hand in all of that. And today I'll be helping Cheryl with facilitating this webinar. Just as house rules, just minimize sound disturbances, please mute yourself and try to keep yourself muted. And if you do have questions, you can type them in chat and we can read those out loud when we do have an opportunity to pause at sections, or you can ask them yourselves during those time. So this is a 2-part series webinar. And so this first one is much more theoretical in terms of the approach of diagram creation. Part 2 on June 10th, which is next week on Wednesday, you'll have much more of a hands-on, more related to your proposal, your diagram that you want to create, and you'll be applying things that you learn from this workshop to there, so that way you can walk away with something that someone else has looked at, or at least giving you feedback on whether or not you picked up the main components of what it means to be a good figure artist. So it is my pleasure to introduce Cheryl McCutchan. She is the founder of Animedia Science. Dr. McCutchan earned her PhD in biology from the University of Colorado Boulder. She is currently a medical editor, writer, illustrator, and animator. So all forms of communications there. And her work can be found in very familiar places like Sapling Learning, which is an online education program, which I actually used when I was in my undergrad. And so, like I said, she's teaching us the fundamentals today. And as they say, a picture is worth 1,000 words, and you can use this to capture your audience instantly. So thank you, Cheryl, for being here. And I'll give you the stage.
Okay, thanks, Joanne, for that introduction. Hi, everybody. So Joanne's already taken my first slide, so I'm not sure I need to go through this. So I was going to do a little introduction of myself, but I think Joanne covered that nicely. The only thing I would add is when I was in graduate school, I went to graduate school in Boulder, Colorado, where I live now. And I did a lot of my work up on the Continental Divide, which is the picture on the right, which is a gorgeous place to work. I have worked with scientists, museums, science communicators to make graphics that explain science. And so I have a few examples here, and I'll have many, many more examples later in the slideshow. Okay, so a general outline of what we're going to do today. I'm just going to give a brief discussion of what research diagrams are and what you can use them for. And then I'll go through the steps for creating a research diagram and use an example that I created for somebody who uses nanotechnology, and I'll get into that more. And then I'll have another example that that I'll be asking for some audience participation on. Okay, so let's go ahead and dig in here. So what is a research diagram? A research diagram explains, and since I'm a scientist and I work mostly with scientists, I always talk about science. So it explains your research with illustrations and text. And so when I create a research diagram, I try to rely heavily on using visuals and visual cues and minimize the text as much as possible so that your audience can just like look through the pictures to understand what you're saying. And often you will also need a caption, a figure caption with your research diagram. So, you know, you balance the illustrations and the text that way. And the main goal of your research diagram is to guide your audience through your research in a logical way. So you're almost telling them a story, and you can think of it as a graphic novel of your research. So you're relying on the visuals and telling a story about your research. And so there on this slide, there are 4 examples of research diagrams I've created for scientists. 2 of these were used in proposals to hopefully get funding, and then 2 were used in publications. Okay, so the purpose of a research diagram, one of the main purposes is to provide your audience with this at a glance view of your research. So they can look at that diagram, get a sense of what you do, and dig in if they decide they want to learn more. And also they're very good for your presentations because a reviewer can just look at that diagram if it's well designed and use that to understand, get a basic understanding of your research. So like, I mean, you can use these research diagrams for publications, proposals. More and more journals are asking for visual or graphical abstracts. And I wonder how many of you have had that experience where a journal either required or recommended a visual or graphical abstract. And you just pop it in the chat box if any of you have experienced that. And also, are there other things that you would use publication or your research diagram for other than some of the things I have listed? You know, the standard things of publications and proposals. The other thing that scientists are using their research diagrams for is social media. It helps them share their research and almost publicize their research to a wide audience. Journals use those research diagrams or graphical abstracts to basically advertise your research or your publication in their journal. Um, and so you can use that as a means to communicate your science and to also drive people to your lab website or your personal science website or your journal article. And then once you have a good diagram, you can repurpose that diagram for different audiences. And you can also repurpose pieces of it for different diagrams. So, you know, if you put in that upfront work, you'll be able to use that diagram in many, many ways. Any other thoughts on that?
We had some
Okay,
people sharing. Oh, sorry.
Oh, so, no, I had said that Joanne was going to take care of all of it, but if I pause, I'm like, oh, I can look at the chat box.
Yeah, I can read it. So we have
Sure.
some comments, and by Tyler says, in chemistry, most journals require a table of contents graphic. Some are good, some are really bad.
True.
True. And Kristen Gabriel says, I have prepared a graphic abstract before, very helpful to capture the audience interests. That's awesome. And then we have Bree saying, I have experience publishing in a journal that required a graphical abstract. I learned the basics of Adobe Illustrator and enjoyed the process of making the research diagram. This is actually a very good point that a lot of us want to do it and would like to express it, but somehow we don't know those, the softwares or the formats people use. So this is a great way to, you know, at some point learning that.
Yeah, yeah. And it's like, pretty good for you for tackling Adobe Illustrator. It is, it is not the easiest app to work with. It's just but it is super powerful. And I'll talk about some of those applications you can use towards the end of the workshop or webinar. Okay, so what I'm going to go through today are the steps that I use to create a research diagram with a scientist. And I've sort of worked these out over the past 12, 15 years to sort of streamline that process. And hopefully these are going to give you like a regular process you can follow to make your research diagrams, and you can come back to when you feel a little bit lost about where to go with all your great scientific information you want to share. So the first step that we're going to talk about is to identify your audience. And I'm going to go through all of these steps with an example too. So you'll get a little bit more information about each step and more practical information. So the first step is to identify your audience. You know, that could be your peers, that could be all biochemists, that could be the general public, or even reviewers who you don't know who they're going to be. And so you have to have some sense of what that audience is when you start designing, because that's going to influence what you put in your diagram. If it's a general public audience or maybe just a biology audience, you want to be careful with the kinds of information and what kind of jargon you use in your diagram and the assumptions you make about your audience. And the next two steps I use are to figure out a main take-home message that is simple and concise, a simple, concise, one-sentence take-home message for your audience. And then from that, develop a few supporting statements that you want to make sure that information is in your diagram. And so that take-home message and those supporting statements are so important because they are going to guide everything you do in making your diagram. And so if you find yourself like starting to pull in some research here and a little here and a little, and it's going outside of your main take-home messages, then you want to step back and say, does that fit? Does that help my audience understand what I'm talking about? And if not, take it back out. You generally want a diagram that is fairly simple and easy to understand and easy to follow. And then once you have your take-home messages, you know your audience, I go and I list everything that needs to be in my diagram. And I'll get to that in step 4. And then, so you can get a better idea of what I'm talking about. And then I try to arrange all those pieces in at least 2 different ways, because I find that the first idea I have is not always the best idea. And you'll see an example of that in the examples I'm going to run through next. Okay, so Joanne, do we want to tackle a question, or do we want to dive in?
Oh, it's more of a question for the future, because you also talk about the
Okay.
file types that are appropriate for different applications, PowerPoint presentations versus publications. What's better, like a .tiff, .jpeg, .ai, etc.? Maybe that's something you want to address now or later. That's up to
Yeah,
you.
well, why don't we save it till the end when I talk about those apps.
Okay, let's go ahead and dive in.
Yeah, but yeah, keep that keep that in your mind. So we do that at the end. Okay, so I'm going to go through an actual research example. I worked with a scientist at Syracuse, and he was submitting a grant proposal. And this was about a nanotechnology subject, a biological nanotechnology subject. And so it was pretty obvious after talking to him who his audience was. I mean, he was going to be talking, the people who were going to read this were the reviewers of the proposal. And he intended it to use for some different audiences also. But he knew that he was going to have reviewers that are going to be scientists. Most likely some of those would be in his field. Some of them would be a little tangential to his field. So, you know, he could include jargon because many of those people were going to be, who read his proposal, had experience in his area of research. Okay, and so talking to this scientist, he gave me a few sort of diagrams he'd put together for, you know, his slides, for his talks, and trying to explain this. And so he and I chatted for a while, and after talking to him, what I came away as the main take-home message was, bacteria are used to produce a variety of nanostructures. And so do you see how simple that statement is? You know, it doesn't go on and on. It's very concise, very simple. And so from that and our continued conversation, I came up with a few other things that needed to be included in that research diagram so that that summary statement was supported. And so in his research, he takes a bacteria and modifies it so that it has special post-transcriptional machinery that then makes these modified polypeptides, and those result in these sort of fantastic nanostructures that can be used for other purposes. And so after talking to him, I needed to make sure that these steps were included. So my summary statements are, bacteria are modified so that they have novel post-transcriptional machinery, and the resulting polypeptides create novel nanostructures. And I could even have a third there that says that those nanostructures are used for different applications. And we'll get to that at the end, because I'll have the final diagram that I created at the end of this example. Are there any questions so far on how you deal with a supporting statement, or how you create that kind of statement? You're going to be given an opportunity to look at one of my diagrams and figure out what those statements might be for that diagram. Okay, I'm going to go ahead and move along then. So the next thing that I do, and this just makes sure that I have all of the objects, the concepts, the processes I need, is I make a list of those, and most of the time I put them on color-coded sticky notes. I mean, if you can see all of my office, there's sticky notes everywhere. I do love sticky notes. And the goal of creating this list is so that I know I've got all the pieces I need to create the diagram, and I also put them in different colors. So I know in this diagram, blue is all of the things. So I've got unmodified bacteria, modified polypeptides, DNA, just a plain polypeptide. And then over to the right, I've got processes in orange. And then normally I have to add a few more things to this as I go along. And so I have all my objects defined. And what I'm going to do next is try and arrange those in a logical fashion. And most of the time I try to create at least 2 arrangements, because sometimes, you know, working through one arrangement, you find something that's not quite right, and then you refine it on the next arrangement. So let's take a look at those, the 2 arrangements I have. Let's see. Okay, so I had information from the scientist, and, you know, I had diagrams from him of the way, and he sort of explained the way he wanted this laid out. And so the first arrangement I tried was similar to one of the example figures that he gave me. And so I had this unmodified bacteria in the center, and then from that, the researchers added special post-transcriptional machinery to bacteria, and the researcher wanted to make sure that the reviewers understood they're not just doing one thing to one bacteria. They're going to try this with many bacteria, and he thought, well, 5 should be good to show that we do different kinds, we add different kinds of molecular machinery, and that gives us different results. So they modify the bacteria, and then the bacteria produce these modified polypeptides, and then those produce modified nanostructures. And so, but the researcher also had this little section up at the top where he wanted these sort of pull-out boxes that had, you know, normal DNA transcription and translation, and then their modified transcription, translation, and post-translation modifications. So when I looked at this first diagram that kind of radiated out, I couldn't quite figure out where to put those. So you could see that the unmodified bacteria did this, and the modified bacteria did this instead. So I went to a second diagram where I started with the modified bacteria at the top, and then moved down through the steps with 5 modified bacteria. And so the researchers create the modified bacteria, which create these modified polypeptides, which create nanostructures. And I already felt like that was working better. And then I realized I could put these little call out boxes to the side, and they would be, you could see them, you could look at them when you wanted to, but they were relatively unobtrusive. So let's look at the final diagram. And so in the end, this is the diagram I ended up producing for the scientist. And there are a whole lot of visual clues that tell your audience what they need to do to understand this. So normally, you know, you're either going to start at the top or I'm not sure which side's the left side for you guys, or you'll start at the left side and read to the right. So I decided to start at the top with that unmodified bacteria. And I think one of the biggest visual cues here is those arrows. Those are telling the researcher's audience exactly where their eye needs to go in order to understand his research. So for me, the most logical thing to do is start with that unmodified bacteria and move down through those steps and follow the arrows. So those arrows are a strong visual cue of what you want your audience to do. There's labels all to the left side, and those tell the audience what's happening in each step and what each structure is. And so they understand this is a process. We're going to go through these steps and we're going to end up with these novel nanostructures.
Oh, it's like if you're sitting at the desk you
and then if you look to the right, then you can see, oh, wait, there's some more information over there. So, you look at it's like the unmodified bacteria. Okay, so this is the way this process is going to work in the unmodified bacteria. And then I can look at the modified bacteria and I can see that something special is added. So there's going to be a unique post-transcriptional modification that's going to result in a unique modified polypeptide. And that's what makes these interesting nanostructures. And then way off to the right on the bottom, we can see some applications for these nanostructures. And I mean, I hope you can see that the stuff to the right is not as important as the stuff to the left and in the center. And that little pull-out box with applications at the bottom is not a particularly important part. I mean, it's going to be useful for the reviewers to see what he hopes to do with these nanostructures. But the main thing that he wanted his audience to see was the steps he was going to take if the research was funded. Any questions about this example?
So not particularly questions about this
Okay.
example,
Okay.
but we do have a question from Emmanuel. These steps seem very straightforward, but these diagrams are going to be iterated over and over again by different people. Can you talk about how you would implement that iteration into your steps?
Yes, certainly. So I would say if normally there's one person who tends to spearhead whatever section of a proposal or paper it is. And so I suspect there's normally one person who is in charge of getting that diagram started. And so I guess there's two ways you could think about doing that. So what I like to do is create a diagram. Even if I don't have all the information, I create, this layout, this initial layout. I don't create the diagram until I've had a chance to talk to the researcher I'm working with. And for you would want to talk to your collaborators long before you put the time in to make this kind of final diagram. So I would probably step back a step. Can I step back a step? And these are relatively crude, of course, and you could certainly recreate something like this in PowerPoint or another drawing program that you like. And what I do is create this general layout before I do any kind of illustration. And I talk to the researcher, or you would talk to your collaborators about, like, is this layout correct? Is there something missing? Is something not connected? And so it's almost like when you have something that's incomplete, you can put it out there and get objective feedback from your collaborators. But to go into a conversation where you may not even have a starting place, it can take a lot longer to work out a diagram by committee than by just throwing something out that you think makes sense and modifying from there. So does that help? Does that make sense? Okay, great, great.
We do have another question from
Mhm.
Andrea. When you're working with a client, do you recommend sharing these various iterations with your client, or do you just show the final diagram?
I normally will show, not the final diagram. I will show them sort of this basic design, not in sticky notes most of the time. Most of the time I'll recreate it in Illustrator, but it'll just be boxes with words and arrows pointing and processes so that the layout is obvious before we start. And then the researcher might come back. And I think I actually, with this one, I think I actually started with that radial diagram. And the researcher was like, well, this doesn't quite fit here. And I don't like this about this. I like this, but not this. And so to have, a starting point that's not, I always start with a draft of the layout. And if something's wrong with that layout, it's actually much more obvious because it's a simple representation of what's going to be the final diagram. Did that make sense? Did I answer that question? Okay, great. Anything else, Joanne?
We have one more question.
Okay.
From, I'm going to butcher your name, I'm sorry, Zhao Ya. I'm wondering, how did you decide the color scheme for this diagram? How do you link colors with meanings and emphasize logic?
Oh man, you are so right. Color is so important. And color is not the easiest thing to work with. So you can see in this diagram, I've chosen sort of a specific color palette. I've chosen sort of this purple and golden scheme, and then that kind of zingy color that's bright green, because I really want the audience to see these are the modifications on the polypeptides. So I could have easily chosen a different color scheme. I could have made the bacteria blue. I could have made the arrows yellow. But you'll notice that I chose two colors that contrast for the main body of this. So I chose purple and yellow, which have a nice contrast. So you don't lose that arrow in the bacteria, and you don't lose it in the background either, because I chose a very light blue to slightly darker blue gradient. And I am looking over here because I'm seeing something about color blindness, which is really, really important to consider. So thank you, Erin. So I can't remember what percentage of the population is colorblind, but I think it's something like 10%. And the most egregious thing you can do with color is put red and green together, because that's the red-green color blindness is fairly common. And so I am very careful with the colors I use when I'm designing a diagram. And I have a special application on my computer that I use to check for colorblind issues. And it's called Color Oracle. And I can mention that again at the end, but it will show you what your diagram looks like in the 3 most common forms of color blindness. And we can talk about that more maybe towards the end of the webinar, but very good point, Erin. Oh, good job. Thanks, Joanne. She's got that link right up there in your chat box for Color Oracle. Okay, so let's see, we'll move on. Okay, so what I would like to do now is show you an example of an infographic that I made. And I want to walk through 3 of these steps with this infographic. And I would like you all to figure out who my audience is for this infographic. And if you could type that up in the chat box, that would be awesome. And you could take a couple, a minute to look at it. Okay, anybody have any ideas who this diagram is intended for? I got one answer up there. Anybody else? Oh yeah. Okay, anybody else disagree? Okay, yeah, keep the answers coming. This is great. It's great to see what you guys think. And so I think, I think many of those answers are correct. I had intended this for a general public audience. So I actually used this on Instagram for an Instagram post that I did with two other scientists. They wrote the post and I was the illustrator. And you're right, it's definitely for the general public. And again, it could be for a more educated public, a general public, because I've got molecular structures on there. Like you can see sort of the right half is much more general and easy to understand. And the bottom left is a little more complicated. So, for people interested in understanding the chemistry of plastic breakdown. And I have also used this for, I've had people request it for their middle school classrooms, middle school and high school classrooms. And I've even had a 5th grade class request this poster for the classroom. So it wouldn't be appropriate, you're right, for younger children. But for a general audience and older kids in school, it's definitely what I intended it for. Or if it's just not your field, but you're interested in learning more about plastics, that's the audience I intended for. Okay, so now let's see if you can figure out the main points I'm trying to get across in this diagram. Like what is, after you look at it a little bit, how would you summarize this in a concise sentence? And what kind of supporting sentences would you come up with? And you can take, a couple minutes to kind of really look through the diagram. Hopefully you can all see it well enough to read it. But if you'd like to try and do that and pop in the chat box, oh, somebody, Rachel. Oh my gosh, I mean, that's so obvious. I'm so happy. Okay, I love the summary statement you've come up with, because that's exactly the statement I was looking for. That plastic breakdown takes a long time, and I'm hoping that yellow arrow that's sliding across the diagram, is the thing that catches your eye first. The title is very obvious, so I'm telling you, this is what you're going to learn about in this diagram. And that yellow, you see the way I use the yellow color against the blue background? It stands out and hopefully is the thing that draws your eye to it first. It also has some of the bigger text, but visually you're drawn to that because it's a bright color against a dark background. And I did that very, very intentionally. Okay, anybody want to try and take a stab at like what else you might learn from this infographic? Okay, so we got a couple supporting statements coming in. Oh gosh, they're going to zoom out of my view quickly. So the fate of nanoplastics is unknown, and I would think that I think of that as more of a smaller point, but that there are two ways for plastic to break down: chemical and physical. And if you look at the diagram, so you go from this arrow, the title, the arrow, and the next largest text in sort of the second brightest area is the text physical breakdown and chemical breakdown. And so I'm trying to use, again, color and the size of font to lead you to those two different methods of plastic breakdown. And then let's see what else we have here. From Tyler, we've got physical breakdown of plastics leads to nanoplastics, which have an unknown fate. That's a very nice supporting statement. Chemical breakdown of plastics leads to leaching of toxic chemicals. And those are very nicely defined supporting statements, Tyler. Okay, and then from Lauren, we have environmental effects of plastic waste on animals, how plastic breakdown occurs chemically, right? And so I've included those animals to show that, like, this is not something that happens in isolation. The animals in those ecosystems are affected by the plastic in their ecosystem, and in some ways we don't understand yet. And then from Miriam, I hope that's right, plastics can be toxic to the environment. So yeah, both chemically and the larger pieces of plastic can be toxic to animals. And I think you guys did a great job pulling out those summary statements and that main take-home message. And my hope in using sort of a simpler example like this is that you can get an sort of an overarching sense of how to do this on something simple before you tackle something complex in your own research. Okay, now I'm going to switch gears a little bit because I'm going to put Joanne on the spot in just a minute. I hope she knows I'm going to put her on the spot, or not. Okay, so I had thought about this for a workshop that I was giving, and I thought, well, let's do kind of a fun example. And so I decided to try and explain grocery shopping during a pandemic, because that seems timely right now. And so for this, again, you can see the color coding I'm using in my initial list. My steps are going to be in yellow, sub-steps are blue, and the objects are orange. And so that helps me keep that that sort of sense of what is what when I start rearranging these. And again, you don't have to use sticky notes for this. Like, if you want to do this in PowerPoint, that'll work fine. If you want to do it in Illustrator, that's fine too. But this is a step that I don't skip. It's especially in complicated diagrams. Okay, so then if we move through this, and I think that some of these are a little bit fuzzy for you guys, and I apologize for that. I came up with 3 possible arrangements for this information, and one just, top to bottom, and then one kind of snakes across the page, and then I just laid the whole thing out in one diagram across the page. And so an important point here is that you may be constrained by, like, the size of a piece of paper or a section of that paper for a proposal, or column width in a scientific publication. And so these are things to consider when you're designing your layout. So if I had a full page, the first 2 are great. If I was going to use just the top part of a page, then I would use the third example. I decided to go with the second example, and here we're going to go to this. And one of the other really important steps when you're using your diagram is to talk to a friend or colleague, a lab mate, and ask them to look at this. And it's especially useful if you can talk to somebody who is a little tangential to your field. So they don't really know your research, and they have the ability to say from an outside perspective, I don't understand this connection here. And I'm just going to check the time really quick. Oh, we were probably, okay, I'm not going to put Joanne on the spot because I want to make sure that we finish all this. I was going to have Joanne walk through this example and explain what I'm talking about in it. And so normally you would have, like when I go back to a researcher, I say, okay, here's a diagram of your research. What doesn't make sense to you in this diagram? What if I got wrong? Or for you, when you design a diagram, you go to your collaborators and say, what is not making sense here? What isn't clear? And then I would also highly recommend going to somebody who does not do the research you do, because some of your reviewers, some of your audience will not do the research you do. And it helps to have that outside perspective on your research. Okay, I'm going to zoom to the end, not zoom, but get to the end here. So we have some time for questions. So my next step after I have that basic overall design of my research diagram, I normally will draw a rough draft that has some of the illustrations and important text I want in. And you don't necessarily have to do that step. If you've got a good, if you've got your diagram laid out and you like the way it is, you can start putting those pictures in. But also remember that in the diagrams I've created, they have a specific style. And I'm trying to use arrows and color and font size for to guide my audience through that research in the simplest way possible. And so the next step, the real next step, once you have a nicely designed draft that's easy to follow and sticks to your main and supporting statements, then you've got to actually get in and make the diagram. And we're not actually going to talk about that in this webinar, because that is a huge topic about how to use programs to create a diagram. But the industry standard for creating illustrations is Adobe Illustrator, and that's what I use on probably 90% of the research diagrams I create. Sometimes universities, you can get that at lower cost, but it's high-end, powerful, and can be anywhere from $30 to $50 a month. So it can be prohibitive. And it's not, it's not necessarily the easiest thing to learn. A cheaper version is Affinity Designer, which is a one-time fee of $50. You just pay for it and you've got Affinity Designer forever. And I think it's actually on sale right now for $25. And it's an alternative to Adobe Illustrator. So it has many of the same functional features as Illustrator, but at a much lower cost. And there are plenty of tutorials online for using Affinity Designer. So that can be a good way to go. If you're totally comfortable in PowerPoint and you know how to use PowerPoint, you can create good research diagrams in PowerPoint. PowerPoint doesn't have the power of Adobe Illustrator or Affinity Designer. It just doesn't do as many things. But you can create a completely functional research diagram in PowerPoint. And more recently, there have been services online that have templates you can use and many assets you can use to create your diagram. So one of these is Mind the Graph, and there's a version of this that's free. And if you need to make a lot of research diagrams, there's a paid version that's $9 a month. And there's also BioRender, and I think it's a little more expensive. The problems that I've heard from researchers with those kinds of online services is they don't have the assets that they need for their research. So they get in there and start designing and then find that, you know, something fairly unique to their research isn't included, like the asset's not there. And so the service isn't that helpful for them. Okay, I'm gonna just pop over here and Joanne, do you want to read me some questions?
Sure.
Move on?
Yes, so we have one from Kylene. Is Affinity Designer easy to learn?
I think that it's fairly similar to Illustrator. And I am almost it's hard for me to judge because I've used Illustrator for literally decades. And once you know one vector program like Illustrator, when you move into another vector program like Affinity Designer, it's pretty easy to switch back and forth. There's with those vector programs, there is a learning curve to using them. But there also are good basic fundamental, like you can find on YouTube tutorials. And I'm actually thinking about trying to create a mini course for Affinity Designer that has like the most important things you need to learn to use Affinity Designer, because there are a range of maybe 10 tools that'll get most of what you want done in either Illustrator or Affinity Designer. Okay, did that help?
Yeah, I agree. I use Illustrator, Photoshop, a lot of the plethora of art tools. And once you sort of understand one, it's easy to migrate between. You just have to know the shortcuts, like the keyboard shortcuts and how to like control your screen. That's pretty much
Oh
like
my
the
gosh,
hardest
yeah.
I mean, you know, I remember my old days when I had no knowledge about what softwares are available, and I would take hours to make these diagrams on PowerPoint. It worked, but I wish I had this webinar before. It would save me time. Thank you.
Well, yeah, and I think the other thing I'm considering is doing, you know, an illustration set of tutorials for PowerPoint, because there are drawing tools that you can use if you don't want to tackle one of these more complex vector programs like Illustrator or Designer. And like, you know, Joanne and I both have said, it's like, oh, there's a few tools that you need, and once you can know those tools, you can use them over and over again.
So another question from Diana, and I'm going to combine this with Isra's, which is, okay. I know you talked a little bit about BioRender. I don't know if you have more of a professional opinion about it, but also if you use icons from BioRender, do you need to give them credit, and how would you go about doing that?
You know, I think with BioRender, you end up with the BioRender logo and company name on your diagram. And so that's, from what I've seen, that's the only credit you have to give, is that when you publish this research diagram, it will have their logo on it. And I think it's appropriate to keep that logo on there so it's not, so you don't give the impression that you drew all of these assets, which would be, you know, a copyright infringement for sure. Did I answer both of those questions?
And I think there is a pricing attached to use of BioRender.
Yes.
So that's something you'll have to look into, especially if you do intend to publish with BioRender.
Yeah, and I haven't looked at that in a while. I mean, at one point it was something like $35 a month to use BioRender, but I haven't checked back in on that price, which I should do.
Yeah, it varies depending on use and what degree of use in terms of individual, educational, lab, institutional. So that's different. Yeah, so another question that we had from before was, can you talk about file types and what they're appropriate for in terms of different applications like PowerPoint presentation versus publications. Which ones should you use in terms of .tiff, .jpeg, .ai?
So what you'll use most often for publications and proposals, what I've seen is they ask for JPEGs. So there's always this balance where you want a good high resolution JPEG, but you don't want it to be enormous, and they can be quite enormous. So you have to balance the resolution with the size. And most of the time when I export something from Illustrator, I export it at several resolutions, and I check to see which is the lowest resolution I can get away with and still have that illustration be clear, because that's going to minimize the file size. You can also export as a PNG. And I know when you publish, there's often a long list of file types that you can import, I believe. But I would say the industry standards are JPEG or PNG, because they tend to make smaller files that are still legible, and everything's clear. PowerPoint, I'm not as sure about PowerPoint because I honestly haven't used PowerPoint that much. Joanne, do you have an idea for that? If you wanted to create a research diagram there and export it?
You can go, I usually just make the image and then select everything, Control C, and then paste it into a Word document, which actually combines everything into a flat image if you paste it as is, or you can export it as a PDF file. And I mean, it'll go into nuances between like vector files versus raster files. So a very quick description of that is a vector file, every pixel or point is an equation. And so when you scale it, it's very easy to scale. There's no pixelation or like the blurry lines that you see sometimes.
And that's a really good point. Really good point, Joanne.
And a raster file is your normal JPEG, PNG types of files. So if you intend to scale things and make it a lot bigger, I would pick a vector file like PDF, which is one of the easiest ones that you sort of find.
Yeah, and lots of there's a lot of support for PDF files too.
Okay. So moving on, we're at 1 o'clock, so if anyone has to head out, there is a survey in the chat. Please go ahead and fill that out. It's just a feedback survey so that we can improve upon this workshop, but we'll continue to answer questions for the next 10 minutes or so, or maybe shorter.
Well, no, that's fine. I just want to emphasize it. It really helps me if you fill out the survey, because then I can improve this, and I can, if something didn't make sense, I know what that is. So it's really, really helpful to me, and I'd really appreciate it. So thank you for all of you who are leaving. We'll keep answering questions.
So another question is, how long does it take for you on average to create one diagram?
It definitely depends on the complexity of the diagram. Actually, you know what? I'm gonna zoom to the next one. I'm not going to talk about the next one yet, but this has just got some diagrams on it. It really depends on the complexity. So the plastics diagram it probably took me 10 hours total, which is pretty fast. The nanotechnology diagram took longer because I was working with the researcher. We were going back and forth about what needed to be done. And I actually had one earlier that I didn't point out that was a glacier diagram. And that researcher and I went back and forth many times, revised that diagram from his original drawing many, many times. And so I would send him the diagram and he would put red all over it, and I'd go back and fix that. So I think that diagram, it was both complex, and the researcher had a lot of input on it. And so that one probably took something more like 15 hours. But some of them, this like a simpler one, like if you look on this, there's the little blue boxes with red arrows. That's quite simple. And so that might take me, I don't know, a couple hours to do.
And another question from Andrea. What applications do you use for animations?
So I have been using, I have the whole Adobe Creative Suite because I use it literally every day. And so I had been using the old program that it used to be called Flash. Now it's called Animate, but I'm starting to use After Effects for animation more. Animate it just has bugs, and there's things it doesn't do very well at all. And so After Effects integrates more easily with other programs in the Adobe Creative Suite. So if I draw something in Illustrator, I can pull it right into After Effects, and which you can't do well in Animate. So yeah, not sure how long Animate's going to last, but you know, if you're going to try and use a animation program, I think there are simpler ones than After Effects, but After Effects has the tools that I need to create animations. And then for 3D animations, I use an open source program called Blender, so like, you know, a blender, and it's free, which is fabulous, and it's very powerful. Again, you know, it's a 3D program, and those take a little bit of time to learn, a lot of time.
Yeah, I use Blender for personal projects as well. It's, I think if you're used to interface of Illustrator, your shortcut keys might get a little wonky there.
Oh my gosh, that is where they came up with their shortcuts. I don't know.
It's strange.
Yeah.
Another question, a comment that we had was the course on Affinity Designer would be very helpful. Definitely something I would be interested in taking from Israel.
Oh, good.
Another question from Zoya. Do we have to obtain Adobe certifications if we want to work as a professional scientific graphic designer?
Oh gosh, no. I am almost, okay, not almost, I am completely self-taught in Illustrator. And what really matters is the illustrations you produce. It really depends more on your portfolio than any kind of certification in Illustrator. I've not, like, it's not even something that I considered doing. I looked at it, I'm like, nah. So your portfolio is the thing that people look at.
Alyssa says, thank you for sharing your sticky note strategy.
I love my sticky notes. Some people don't like it, but it's one of my favorite techniques. So you're welcome.
Another question from Zoya. Would it be possible to employ coding or machine learning skills to enhance efficiency in graphic designing?
Wow. I'm not sure I have an answer for that because I'm not very good at coding. I mean, I code some stuff in Animate to get particular animations to occur and stop and things like that. I do not know the answer to that question. I would love to hear your thoughts on it.
I think it'd be very interesting, especially once you sort of do, I forget what it's called, but when you're connecting steps in between movement of images and whatnot.
Mm-hmm.
But I don't know, I'm not sure to what extent, because I've done everything quite manually.
Well, in a program, if you're using something like After Effects, I mean, there's not a lot of coding involved there. It does almost all of the hard work for you. So yeah, I'm not sure the answer to that question.
That'd be a really interesting thing to explore. So yeah, if you feel like there are any niches that need improvement, I think there's definitely like Adobe Illustrator, Adobe company, like they've been around for so long, and that's sort of the standard of the industry. So if you can improve upon them, that would be a game changer.
Yeah.
So one last question from Bree. What tools did you use to learn Illustrator? Is it YouTube or something else?
Well, I would say a variety. When I was first working with Illustrator and really getting into illustration, there was a service called lynda.com that had fabulous courses you could take. And it was something like $35 a month. And it was totally worth it because the courses were exceptional. And I learned a ton from those courses. Those courses are now on LinkedIn Learning, and I believe you can still find the same courses, the same quality, but LinkedIn bought Lynda, and so now that material is there. You can find things on YouTube, absolutely. The only problem with using YouTube is it can be a little bit haphazard and random where you find things. Like if you need to learn how to use the pen tool, go onto YouTube and search pen tool Adobe Illustrator, because you will come up with many videos that talk about that. And also the Adobe site has basically a manual for every program. So again, if you wanted to figure out how to use the pen tool and you would rather like read about it and, you know, have their screenshots there to look at, you can go to Adobe and search. I mean, you can even just search Adobe Illustrator pen tool for that example.
Yeah, so do you want to go ahead and just wrap up for the next couple of minutes?
Sure. So if you get to the point where you're stumped on how to draw something, you can hire a professional like me or a graphic designer with experience with science. I mean, what I found is if just a graphic designer often doesn't have the background to work easily with scientific concepts. That's, you know, just my little tip there. And so next week is the second part of this webinar series. And in this, participants will actually be making their own, you know, sticky note research diagrams, defining their audience, defining their main take-home points, etc. So you can talk to Joanne about this. It'll be next Wednesday at the same time, but it'll be an hour and a half instead. And then I want to thank you all for showing up. I hope that you've learned something, and I hope that this has helped in the way that you look at designing your research diagram for your research. And if you would like to email me with questions, my email address is cheryl@animediascience.com. And I'm happy to, you know, you want to send me an email, I will answer your questions to the best of my ability. You can also find me on Twitter at Dr. Cheryl Draws. And I'm starting to put more information about designing research diagrams on my Twitter account. And then if you'd like to connect with me, you can find me at Cheryl McCutchan, PhD on LinkedIn. And thank you all so much for taking the time out of your busy day to learn about designing research diagrams.
Yes, thank you, Cheryl.
You're welcome.